Hey, Owen.
Hi, Steve. How are you?
Good. How are you doing?
Good. Good.
Good. So I think I've set it up so you are able to share your screen whenever you want to do that, but no rush.
Great. Well, why don't we test it now and make sure it works? Because
That's
that's
a good
that's
idea.
that's always the thing. Let's see. Let me just give it a shot here. All right.
Okay, looks good. Now I have to figure out how to get my video to go away. I'm gonna try.
Able to see the slides changing and all that.
So is it a busy time of year for your CCS work?
Yeah, it never seems like it isn't. We're in the field a lot now, both institutionally because right whale season, and then, even some of our other, like I'm on the water and like little small open boats right now. Like it doesn't stop. Um, but every year is a little different. It really depends on what projects we have going. And we were out in Nauset Marsh last week working, and
Great.
they're getting those calm days still here and there.
Right. I know. Just enough.
Yep. Yeah. Barely enough. Exactly.
It's good. Well, welcome everyone. We're getting a lot of people joining here. We'll give it a few more minutes. Welcome everyone who's joining us here. We'll just give it another minute or two before we kick things off. But we've got a lot of people on the call, so this is great to see. Thank you, Kevin. All right, well, it's 6 o'clock here. I'm sure we'll have people continue to sign on, but I've got a number of introductory things to get through here, so why don't I kick it off. So thank you all for joining this webinar this evening. I'm Steve O'Grady. I'm the Executive Director of the Orleans Conservation Trust. And in recognition of the broader audience that we're able to get through these online lectures, I would like to just say a few quick words about OCT for anyone who may not be familiar. So the Orleans Conservation Trust is a land trust that was founded in 1970 and is the oldest continuously operating land trust on Cape Cod. So keep that in mind next time you hear someone from the Chatham Conservation Foundation talk about how long they've been around. You can say, yeah, but OCT was continuously operating that whole time. So OCT has preserved over 640 acres and continues to acquire land by gift and purchase, all of which land is preserved in perpetuity. We host monthly guided walks in Orleans, and we sponsor this wonderful lecture series that you're about to enjoy. We have 2.5 staff and a very engaged volunteer board of 13 trustees. All of our work is made possible with your support. We really, really appreciate the generosity of the Orleans community, and that has enabled us to be around for 50 years. And this is the part where I post the donate link in the chat pod for anyone who may have missed your donation in 2020 particularly. But thanks all for being here. And let me quickly go over the basics of how this Zoom webinar will work, although I know many of us are all too familiar with this platform at this point. So as an attendee, your camera is off and you don't have the ability to turn it on. Though we like to be able to see each other during Zoom calls, we just have too many people who are tuning in for this webinar. And Owen's a big draw, as we know. So, but that said, you will be able to ask questions using the Q&A feature that you should be able to see at the bottom of your screen. And you can choose to ask those questions anonymously if you'd like. There's a box that you should see to check to be able to ask questions anonymously if you want to do that. And then you should also, I hope, have the ability to use the chat feature. I tried to set this up this way, and I've seen some come in, and I think you can chat with individual other attendees. So you should be able to see, hopefully, who else is on the call if you'd like to chat with someone in particular, and then also just chat to the whole group. If you have questions that are coming up as Owen is presenting, please just put those in the Q&A, and at the end of the presentation, we will walk through those together, and Owen will do his best to get all of those answered. Let's see, I think that's it as far as logistics. So let me introduce our guest that we're so lucky to have, Owen Nichols. Owen is the Director of Marine Fisheries Research at the Center for Coastal Studies, where he's been since 2008 in that role. He conducts research in collaboration with Cape Cod fishermen and shellfish farmers. His primary interests include distributional ecology, fisheries oceanography, marine mammal and fisheries interactions, and ecosystem-based fisheries management. Owen is a PhD candidate at UMass Dartmouth School for Marine Science and Technology. He's a guest investigator at the Woods Hole Oceanographic Institution, and he's also an adjunct professor at the Mass Maritime Academy. With all the time left on his hands, Owen also recently became a founding board member for the newly formed Sips Island Trust. And this last commitment ties in nicely with his presentation for OCT. Obviously, OCT is a land trust, and Owen's studies are marine focused. But I think anyone who studies marine ecosystems, and especially estuarine ecosystems, can tell you that it's very important what happens in the land that surrounds those estuaries. So Pleasant Bay watershed, Nauset estuary watershed. So OCT's work and other neighboring land trusts work to preserve land in those watersheds has a profound impact on the water quality and what we see there. Owen holds a BA in Marine Affairs from the University of Rhode Island and a Master's in Science in Marine Science and Technology from UMass Dartmouth. So without further ado, thank you, Owen, for joining us, and I'll turn it over to you. And Owen's going to screen share here, I believe.
Great. Yeah, thank you so much, Steve, and to the Trust for having me tonight. Yeah, I'll put up my slides here momentarily. I hope you'll forgive the big headphones. Noise canceling headsets are the only way to co-work at home with other folks. So it is what it is in these times. But yeah, I'm looking forward to sharing some of our work with you. And, you know, I think you're absolutely right, Steve, that there's so many connections between, you know, the marine and estuarine work we do and the upland conservation work that you all do. You know, and in many cases, you know, we're seeing more and more coastal properties held by different trusts, and in some cases, island properties, as you alluded to. You know, and I think a lot of the same principles apply that we'll be talking about tonight. So I'll go ahead and share my screen here.
Well, you're not getting your toes wet with land conservation, you're getting them dry.
That's right. All right. Give me a nod, Steve, if you guys can see my slides there.
Yes, that all looks good.
Wonderful, thank you. So before I get into this here, you know, I first of all want to acknowledge the fact that the other hat that I wear at the Center for Coastal Studies is I coordinate our interdisciplinary research. And what that essentially means is that in addition to doing our fisheries work, I also try to bring together a lot of the other really diverse expertise that we have at the Center for Coastal Studies in order to address sort of complicated natural resource questions We have a geology department, we have folks who do water quality. And so the work that I'm going to present to you here represents work done by a lot of people. I also do want to acknowledge the fishing community. As Steve mentioned, I do a lot of work in partnership with the fishing community, and a lot of their expertise, their insight, the motivation that those folks have to understand our coastal habitats goes into this work as well. And we do a lot of this work, in partnership or under advisement or in consultation with our local fishing communities. Our fishermen know these waters so well. And to that end, in this title slide here, you can see Ted Lucas, who works with us at the Center for Coastal Studies, but has decades of experience as a commercial fisherman. And that's the kind of expertise that he brings to the table for us as he runs our research boats, but does a lot more than that, really provides a lot of expertise in the work that we do. So the way that I want to go through this, I really kind of want to get right into the work that we're doing, where we're doing it, what we're doing, what we're seeing. And then I want to kind of scale back up to why we're doing it, some of the broader context of ecosystem assessment and the need for long-term monitoring. But without further ado here, let's go ahead and get our toes wet here. We'll start right in Pleasant Bay with some of the work that we do there. And Pleasant Bay, as many of you know, it's a really fascinating, really interesting system. It's surrounded by 4 different towns, including much of Orleans. It's a really dynamic coastal habitat. The shorelines are constantly changing. It's a place that has a very long history of natural resource harvesting, shellfish and finfish harvesting. But it's a really fascinating, really complicated, and really dynamic ecosystem. As many of you are familiar with, the shoreline, the barrier beach system along the outside is constantly changing, constantly in flux. And with that comes a lot of natural change in the system itself in terms of water flow. And so it's a really fascinating place to work. It's a really interesting place. Again, it's constantly in a state of flux. And it's also a place, like many of our coastal habitats, that are profoundly affected by human activities. We have a lot of concerns about water quality, fish populations, and all of these different things. And way back in the 1960s, the State Division of Marine Fisheries, and I'm going to take a very much sort of fisheries-centric approach to this, and I'll start with some of the historical work done by the state. The state started this series of estuarine studies that were done all around Massachusetts, looking at some of the larger coastal systems. One of those systems, the marine resource studies they did, was focused on Pleasant Bay. And so they did this work back in 1965 and 1966. And this is a really comprehensive set of observations. I think you can get this report on the Pleasant Bay Alliance website, among other places. And they did a lot of very fisheries-focused work, trawl surveys, seine sampling, so using different types of sampling gears to assess what was out there in terms of living marine resources, generally with a focus on fish and shellfish, things of sort of food importance, cultural importance to us. But one of the key recommendations of this big study was that within 10 years, another study should be done. And no work had been done for 10 years, 20 years, 30 years, 40 years, 50 years, until we ended up going back out into the field and doing that. And the reason we went back and repeated a lot of the work that they did was in large part because of the really great work by the Pleasant Bay Alliance and the natural resource management plan that the 4 towns through the Alliance have for Pleasant Bay as a system. And this was back in 2013. Their resource management plan really noted that there had been a lot of changes in finfish and shellfish abundance, species composition, fishing activity since that work had been done in the 60s. And one of the key recommendations was that surveys ought to be done of Pleasant Bay's fisheries habitat, and really to develop and conduct a long-term monitoring program. So this is where we really got interested. It's like, okay, this is a study that has broad interest. We did a lot of outreach to the fishing and shellfishing communities and the natural resource managers, and everyone really seemed on board with this. So we put together a proposal to the Friends of Pleasant Bay. Um, and this proposal was in 3 parts. It included really comprehensive benthic habitat mapping, which had also been partially supported by the National Park Service, by the Cape Cod National Seashore. Um, the study that I led, looking at distribution and abundance of shellfish and finfish. And we also did a lot of work looking at seal populations in and around Pleasant Bay. So this is a really comprehensive look at an ecosystem on Cape Cod, and really looking at this system in as many different ways as we could. And this really kind of set the stage for a lot of the other work that we were doing. Again, I'm going to focus mostly on the fisheries component at first, and then I'll kind of scale back upwards a little bit. We're going to talk a lot about sort of general species of interest, natural history, and again, sort of with a fish and fisheries focus. We started our work in Pleasant Bay in 2014 with a pilot study also funded by the Friends of Pleasant Bay. And this was a study very much focused on lobsters. There's a keen interest on behalf of Charlie Beggs, who ran a mooring company and was also a shellfish harvester in Pleasant Bay for a long time. And he was really keenly interested in the fact that he noticed that there were a lot of juvenile lobsters on the mooring blocks every fall. So again, very much in line with the kind of work that we do, listening to the expertise and the knowledge of professionals working on the water. We went out, did a focus study, and Charlie was kind enough to lend his expertise in his mooring barge so we could go out and put these 200-plus-pound cages full of stone wrapped in mesh out onto the seafloor in Pleasant Bay. And this is one of many different ways you can sample marine habitats. We left these out for an entire summer, and we'll let you know what we found here shortly. This is just one of several tools and part of a pilot study that we did. But with that, we began a full-fledged study with that support from the Friends. And we went back out there starting in 2015 in a really much more comprehensive way, trying to look at fish populations and shellfish populations in as many different ways as we could. And one of the tools that we have for that is seine sampling. This is a 60-foot long, essentially a bait net. They use these down south to harvest commercially. But for us here, it's a sampling tool, and it's very much consistent with what the state used in their work in the 1960s. So again, trying to replicate the work that was done in the past in a standardized, replicable way going forward. And we did this mostly in the northern part of the system in 2 sites that the state had surveyed in the 1960s. And we did the bulk of our sort of deeper water work from a small boat. This was the pride of our fleet at the time, the research vessel Shackleton. You can see her tied up here at Nauset East. Scott Noakes and his folks there really helped us out a lot in giving us a place to work from. And we did a whole lot of survey work down in Pleasant Bay. And what we were doing is, if you look on the right-hand side, you can see all of those green dots. Those are all benthic habitat stations. So where we took a series of grab samples from the bottom to come up with information about what lived in the sediments, what type of sediment there was on the bottom. So literally kind of working from the bottom up here, we went out to those benthic stations where those grab samples were taken. And we went back over as many of them as we could reach with our trawl and dredge gear that I'll show you. We went back over them throughout the year and surveyed as many stations as we could, as much as we could do with weather and tide and wind and so forth. The tools that we used working from that smaller boat, again, kind of relying on the expertise of commercial fishermen, is really not much better gear for sampling sort of macrobenthos, in other words, the bigger invertebrates and other animals that live on the seafloor, than a bay scallop dredge. We used a very lightweight one without a cutting bar so that it would make a minimal impact on the seafloor. And we also used a 30-foot sweep otter trawl. It's essentially a small trawl net that's towed behind the boat. Again, relatively minimal bottom impact from this net. And it's exactly the same net that the state had used in previous studies. So again, trying to replicate the methods that they used in as many ways as possible to be consistent as possible. We also used a variety of other gears. Here we have some small ventless lobster traps, again, very similar to commercial gear, but smaller mesh and without an escape vent. again, really kind of hitting this system with as many different types of gears as we can and trying to take a really comprehensive look at what was living out there. We'll get into some of the results here from that work in Pleasant Bay. So first thing we did in fall of 2014 was haul back our collectors that had been sitting here all summer. So remember those cages full of rocks surrounded by mesh. You can see the fine mesh at the bottom there. And the reason you fill these cages with rocks is because it creates a lot of interstitial habitat, these spaces in between the rocks that the fish and invertebrates will go in. So once you carefully remove the rocks from the cage, this is what you see there on the left-hand side. You can see all of these different types of fish laying there. And of course, we did get a lot of these little young of the year lobsters right in the middle there. Those are essentially less than a year old sort of post-settlement lobsters. So we did see a lot of those, and actually the index that we had in there, sort of the number of lobsters per square meter, was actually higher than in many other areas in the state, right there in Pleasant Bay. But what was also really interesting, we got excited about the lobster settlement that we saw, but also the variety of different fish species. If you look on the right-hand side, you can see a couple of the more exotic specimens we got in these collectors. And these are both species that are found much more commonly to the south. They're reef fish. The top one is a juvenile snowy grouper. The bottom one is a spot-fin butterfly fish. And this really promoted a whole separate interest in these sort of tropical fish, working with a master's student up at UMass Boston who accompanied us on many of our research trips. He calls these sort of wayward little juvenile fish Gulf Stream orphans because they come up in these warm water currents, probably as larvae or small juveniles, and spend the summer in our warmer estuaries. But after that, we don't really know what happens to them. So we could talk about that more later on, but it really kind of promoted a broader interest in what else might be living out there in the fish community. So we got right to work the next year with our seine surveys first. And when we were seining in some of those different parts of Pleasant Bay, we saw a lot of the things that you might expect to see. Lots of bait of all different kinds. Your typical minnows, your mummy chugs, your killifish. That's a striped killifish there at the left, the larger photograph. Mummy chugs at the top right. Schools of menhaden or pogies. And some more exotic looking species as well, like all the way towards the bottom right here, that's a needlefish. Those are great food for striped bass. One of the more interesting species we saw in the seines, needlefish, so named for that long snout there. And if you look closely, it's hard to see here, but they actually have really sharp, jagged little teeth there. And some of the more abundant species that we saw are the silver sides. And just to give you a sense for what sampling like this is like, when you pull that net in, sometimes you get a lot of fish. And just as an example, we saw one haul of that net to shore, 2,715 silver sides that we counted in that net. And it's a challenge to very quickly count and release those fish while also measuring as many of them as you can. When we get into our trawl and dredge surveys, I'll show you sort of 3 different spots in Pleasant Bay, just to give you a sense of the different kinds of animals that are out there. First of all, in terms of fish species, and this is a spot just off the property there at Pawa Pond, some conservation land there. And just off of that, just off the big rock there at Pawa, we were towing and dredging and seeing all kinds of different animals. The fish right here in the middle is an Atlantic tomcod. These guys are well known to spawn in Pleasant Bay. They're cousins of the Atlantic cod that many of you are more familiar with. We did see lots of bay scallops. It's rare that we saw many large scallops, but in the first year of our study, we did see a lot of seed, like you can see at the top right here. And then this little guy at the bottom right, something that we found often around eelgrass, that's a lumpfish. You can see why they're called a lumpfish. They have a little suction disc on their ventral surface on their underside, and they'll stick onto buoys and eelgrass and your finger and anything else they can grab onto. They love structure. And always crabs, lots of crabs. That spot off Pawa, we saw some of the largest spider crabs we encountered. That's one of our interns, Emily, at the top right, with 2 large male spider crabs. They're somewhat distantly related to king crabs, and you can see the similarity there with those large claws. Not a lot of meat in there, though. Um, Bottom right, all the way to the right-hand side, you can see a green crab with eggs here. Now, green crabs are an introduced species. They're one that we're actually fairly concerned about, as they're fairly destructive to shellfish populations and marsh habitats. And then in the middle here in the bottom, at first glance, you might think this is just one blue crab, or rather two blue crabs here. But what you're seeing is this is a blue crab that we actually caught in the act of shedding its shell. So crabs need to shed their shells to grow. So what you can see here, this is the old shell of this beautiful blue crab. And this is the new shell that's coming out. It's still quite soft. If any of you have ever had soft shell crab, that's one of the things you're most likely eating is a soft shell blue crab. And very popular down in Maryland, for example. But you can see already as this soft shell is taking in water, it's expanding a little bit. It's already a little bit wider than that old shell, and it will widen a little bit further as it hardens up. But anyways, species in some cases of commercial or recreational importance, and in some cases also species of concern like these green crabs. When we get down into some of the coastal ponds, a little bit different picture. This is Crow's Pond down in Chatham. Unfortunately, the seafloor inside this pond, we had a lot of tunicates or sea squirts and a lot of really anoxic sediments in there. Some of the work done by the Pleasant Bay Alliance looking at water quality over the last a decade or more. Some of these coastal ponds have relatively poor water quality, low dissolved oxygen. And one of the things we did see a lot in there were squid, which is a nice thing to see. Squid are an important forage species for a lot of different animals, and also happen to be one of my favorite critters. And we did also see squid eggs, so we knew they were spawning in places like Crow's Pond. But unfortunately, you'll see, you can tell by the coloration here, that kind of milkiness to them and the blackness, that a lot of these eggs were actually getting smothered. A lack of oxygen down there on the seafloor were causing these eggs essentially to rot. On a more positive note, on the bottom right, this is a relatively poorly understood species of goby that I'd never seen before. These are called seaboard gobies. And Pleasant Bay at the time represented one of the furthest north and east that we'd seen this species. And this is one of those things where if we were kind of using normal sampling gear, just towing a net, we wouldn't have seen these. But by using different gears like that small dredge, we're picking up a lot of bottom structure in our short dredge tows. And we found a lot of these little gobies inside structures like shells. And they also seem to really like empty beer cans. We found a lot of them in old beer cans. Another interesting thing that we found in areas around Crow's Pond and Eastward Ho is this strange looking benthic organism. They're perfectly spherical and covered in pock marks. And imagine you can guess what those are. And closer look, you can see Titleist written on a few of them. But again, a whole diversity of life here in Pleasant Bay. When you get down towards one of the inlets, again, you're now in a different habitat. Again, sandy bottom, mussel beds, lots of these juvenile mussels spread out across the sand. And again, diversity of fish in particular. This species at top center is winter flounder or blackback. That's one of the historically most important commercial species in Pleasant Bay. There used to be a small trawl fishery for those in Pleasant Bay, and we did see quite a number of them down near the inlet. All small ones though, and we'll come back to that. And a lot of forage fish like these sand lance here that we generally associate with more open water. Again, sand lance, one of those really important forage species, bait essentially, species that whales and larger fish and seals and other creatures really rely on for calories and energy. And again, lots of crabs. Here's another one of our interns at the time, Tim Famulari. And he's recognizing that he's now going to have to extract and help me measure every one of those rock crabs that literally plugging up to the top of that lobster trap there, one of our ventless traps. So again, a lot of differences from site to site in species composition, both in the fish community and in the invertebrate community, and just an overall sense of the diversity of life out there in Pleasant Bay. So we got an enormous amount of data from the system. From our systematic surveys, we counted over 14,000 different organisms. You can see how those compared. We actually caught a lot more things in our seine sets than we did in our trawl and dredges. That actually makes sense because we have relatively short bottom times for those mobile gears like the trawl and the dredge, wanting to make a minimal impact on the bottom. And a lot of the species that you capture in a seine tend to be schooling fish that travel in large numbers. We were able to do surveys fairly consistently throughout the year, and a lot of the sort of seasonal patterns that you see. So the abundance as it goes across the year from January to December, the patterns in that seasonality of a lot of the different species were relatively similar to what was seen in the 1960s and some of the other regional studies that have been done. One of the things that was really notable about Pleasant Bay, and this is fairly typical for habitats like this, but a lot of the fish species that we saw were only represented by juveniles or essentially young of the year, so fish in their first year of life. All of the winter flounder that we saw based on length would have been juveniles. No adults were seen, and that's something that we're seeing throughout the region where we think the large flounder generally don't come inshore to spawn anymore, but the juveniles still use it at places like Pleasant Bay as a sort of nursery habitat. And to the right there, that's actually a more pelagic species. Those are not river herring, but those are sea herring that have come inshore in their very first few months of life to use Pleasant Bay as a essentially we'd call it a nursery habitat, very early life habitat for these. You can see this is right at the bottom here. This is sort of what you call a post-metamorphosis smolt here. This little sea herring, you can still see through it. You can see the lines on the length board behind it here. And because we got a fairly wide range of sizes here in the trawl, you can see the progression just in this one toe. And I think this was in, I forget, well, Lonnie's Pond actually. In Orleans, and you can see the progression here, but that's just one toe, and all of these fish are days to weeks apart in age. Again, a diverse assemblage of marine animals and spawning in nursery habitat for a variety of different species. You're seeing species use the bay for their entire life cycles. So here you see that's a tiny little juvenile squid on the palm of my hand there. That's probably less than an inch long. And this is a small adult squid here. But again, wide variety of size classes in some of these species. And of course, we saw the eggs there too. So we know that they're spending almost their entire life cycle in some cases within Pleasant Bay. In general, when we look at what we found in Pleasant Bay, we used more sampling methods and we covered greater area than what the Division of Marine Fisheries did in the 1960s. And, consequently, we saw a greater species diversity. When you subtract all of the invertebrate species that we saw and compare more directly, species diversity wasn't that much different. It looked like in some cases it was a little bit greater at some sites in the 1960s than it was today. And more notably for us in our study, we saw that there was differences in diversity and abundance between years. And that's a point that I'll circle back to because it is an important one. But the reality is in this fish species here to the right, that's an Atlantic moonfish. That's another one of those tropical species of real interest to us. But really, these are just, even though this is an enormous amount of data and an enormous amount of work over the span of a year, it's still just a snapshot in time. Both the work that the division did in the 1960s and the work that we did in 2015 and 2016, and again, a little bit in 2017, these are just snapshots in time. And, without the context of looking at the long-term changes, a lot of the changes that we see are cyclical. Comparing 2 points so far apart in time is interesting, but it doesn't tell you a lot. You really need long-term monitoring to track changes over time in these systems. And that's a theme that I'll come back to a few times tonight. I want to move a little bit north along the coast. Again, still staying in the town of Orleans, at least in part. In this case, though, this is work that's funded by the town of Eastham. And this is part of another large, very comprehensive study, really focused on the dynamic changes of that barrier beach system. Our marine geology and seafloor mapping groups are really taking a hard look at that barrier system, how it changes over time, doing 3D mapping of that system with drones. I come back into this project much as I did in the Pleasant Bay project, working with the seafloor mapping group, looking at benthic habitat, and then focusing on the fish communities in the system. And so that'll be my focus again tonight. But again, part of a much broader study, very broad in scope, to really understand the system as a whole. So much as we did in Pleasant Bay, we're revisiting a lot of benthic habitat sampling sites. Here's an example of one of the grabs that we use to take samples on the seafloor. A lot of these, all of these red dots are places where we did work back in 2014, supported by the National Park Service. We're going back out and revisiting some of those sites, and we're doing it at a very high temporal frequency. So doing it essentially every month to every other month, and really looking at fine scale changes in the species community, in essentially the worms and other sort of very small organisms that live in the sediment, in the sand. So to look at change on a very fine scale. And we're going back out and doing a lot of the same survey work, the trawl and dredge work. There was some work done out there in the 1980s by Ken Abel and his group down at Rutgers, working with the Park Service. But we're adding a few tricks to our repertoire of different types of sampling gears. One of the things that we didn't do well in Pleasant Bay was sample some of the faster, larger, more mobile fish. We tried gill netting to sample bass and bluefish, and it really did not go well in Pleasant Bay. So we're trying a new method here, new to us anyway, but a well-established method. It's called baited remote underwater video, or BRUV. You put a pyramid down with a bait cage in a set of video cameras. You can often use 2 cameras. You can do stereoscopic measurements. You can put laser reference points there. You can measure fish. You can count fish. And so it'll give us a good sense of what's down there for some of these larger, faster, more mobile species that we wouldn't otherwise be able to capture with our slow-moving trawl net or by other fixed gears. And if you see us out there in Nauset, we're doing field work right now. That's the boat you'll see out there. We were just out last week in these occasional calm days here. So if you see us out on the water, that's the research vessel Kingfisher. That's the boat that we're using now for a lot of our coastal sampling work. So if you see us out there, give us a wave. Field work out there is ongoing. There's a lot of life out there, even in these winter months. And on the right-hand side, that's another introduced species. That's a European oyster that we captured in the dredge sampling that we're doing in Nauset. And to the left-hand side, that is a tiny little blue crab doing what blue crabs do really well, which is pinching my finger. Even that tiny little one there is got a pretty good little grip on my finger there. So this work has just started. We don't really have any summary tables yet or anything like that. We've just gotten on the water in November and only done a few days of sampling so far. So stay tuned on that. You'll see us out in Town Cove and Nauset Marsh for another year. Switching gears again, I want to talk about just a couple of the other places that we're working because again, there's a lot of common threads, a lot of commonalities between these systems and also why we're studying them. So we're doing some work, very early work in Wellfleet Harbor, again, along the same lines, the site of another one of these comprehensive studies by the state back in the 1960s. Also hasn't been duplicated since, much like Pleasant Bay. We're building on some of the early seafloor mapping work we did there. Our fish sampling that we're doing in Pleasant Bay is a little bit limited, or sorry, in Wellfleet Harbor is a little bit limited so far. We're hoping to expand this study. We're seeking funds to make this a much larger, more comprehensive study, as we've seen in Pleasant Bay and Nauset. And I wanted to put some slides up from Wellfleet just really quickly, not because they're dramatically different in terms of the species communities and things like that, but I want to show you what field work looks like in the socially distanced world of 2020 here in 2021 with the challenges of COVID. You can't get much more socially distanced than working at opposite ends of our 60-foot beach seine there. Those are a couple of our interns and volunteers in the field doing seine sampling in Wellfleet Harbor. And just showing some community participation here. We've got a family group of volunteers, mother and her kids working together, volunteering along with our intern and volunteer, all pecking the net together, dealing with those large numbers of fish that we encounter from time to time. In this case, those Atlantic silver sides that are so abundant when we seine. We did see a number of small bluefish in our sampling studies as well. It's one of our volunteers, and James from AmeriCorps Cape Cod helping us out in the field as well. One of the exciting things that we are seeing in Wellfleet Harbor, as we've seen in some other places, is lots of Atlantic menhaden. That's a really important species. Many argue for water quality because of the way they filter the water as they feed. And we saw over 1,500 Atlantic menhaden in a single seine haul there. And these fish are a really important part of the system for a variety of different reasons. But what's notable is some of the same sampling work that was done in the 60s didn't see any of these fish. So we're exceptionally excited to see those. I think that's all I want to say about the work that we're doing in Wellfleet. And now I want to scale back upwards a little bit and start thinking about why we're doing this. Why is this important? What is the purpose of all this? And, again, right back to what I said at the beginning, all of these systems are facing a variety of different stressors, things that are affecting the system. Some of them are natural, some of them are anthropogenic or caused by humans. We're dealing with the effects of climate change on a lot of different scales. The Gulf of Maine, north of Cape Cod, is warming really rapidly. We're seeing a lot of unusual species, and this is a very Wellfleet-centric slide, but you could argue that a lot of these things are happening in all of the different systems we've talked about. There's some unusual fish that came up at Wellfleet a couple of years ago. Some folks who were mackerel fishing were catching a lot of these banded rudder fish, relatively unusual species to catch on hook and line in Wellfleet Harbor in the past. The Herring River Restoration Project that's going on in Wellfleet right now, very typical of some of the other restoration projects going around in our coastal habitats. I know folks in the conservation community are very familiar with some of these projects. Of course, dredging, a big issue in a lot of these different places, certainly going on in Wellfleet Harbor. And of course, our finfish and shellfish harvesting, particularly shellfish, both aquaculture, culturing shellfish, and wild harvest in so many of these systems. So with all of these different things going on, it's really important to establish baseline information, so get a really good comprehensive snapshot, but also to really start developing a framework for how we're going to assess changes to understand the relative impacts of all of these different stressors over time. And also it's important to make sure that we're really asking the right questions. There's so much we could do. And so to try to boil it down to some of the more important relationships between animals and their habitat, and making sure we're asking the right questions of the system and of our data. So when we do that, when we take this sort of broad scale ecosystem assessment approach, we're really looking at things at a variety of scales and space and time. We pull in as many different data sets as we can from our work, and in many cases working with others as well. Again, kind of looking back at Pleasant Bay, this is an example of some of the sonar data that we have for the system. So our seafloor mapping group going in there with high frequency sonar and taking bathymetric measurements of the seafloor, so looking at the underwater topography, but also taking very high resolution photographs with a high frequency side scan where you can see things like this eelgrass patch here. That's a mooring block in the middle with a mooring scar around it from the chain. You can see that even showing up in the lower resolution bathymetry data. So, this is an example of the types of data that we can pull into these assessments. We have those benthic grab samples where we're taking samples of the sediment type at the bottom, the organisms that live in that sediment, and we can start to pull all of that information together with the types of data that I've talked about more today, looking at sort of the larger animals, the macroinvertebrates and the fish. And we can start looking statistically at the relationships between those species communities and their habitat, what animals are associated together most often and with what habitat type. And we can look at basic things like diversity from site to site and start understanding which areas might be most important and which relationships might be most important between animals and their habitats. And we're really trying to consider those results in a broader ecosystem context and to try to present that information to the world in that way. If you want to learn more about the work that we've done in Pleasant Bay, a couple of years ago, we put out a very comprehensive report. It's available on our website. coastalstudies.org/interdisciplinary, where you can find the reports that we did in Pleasant Bay. And we've also undertaken a really strong effort to put this information out into the scientific literature as well. We recently, in 2020, worked to publish a special issue of Northeastern Naturalist. So that's a peer-reviewed scientific journal. So trying to get this information out into a whole different group of audiences, your scientific audiences, your natural resource managers, the general public. We want this information to be useful and applicable in as many different ways as possible. Now, I do want to kind of come back to this concept of baseline data and the importance of monitoring. It's great to take these really comprehensive snapshots in space and time. It's really important to do that. But we really need long-term monitoring data. And a good example of that is, as we look at how what we do affects the system, the top two photographs are that incredible undertaking, the Muddy Creek Restoration Project down in Pleasant Bay. And we went out there, we were working out there in 2015, 2016, 2017. We weren't doing work focused on Muddy Creek. But when we went back out in 2017, after the bridge had been finished and the tidal flow had changed, we actually noticed, even in a small number of samples, a really different finfish community outside of Muddy Creek. We saw scup, butterfish, sea bass, a whole different species community than we'd seen in the past 2 years. And that was really interesting. But again, without sort of long-term data, is that just an anomaly because of warm temperature, different species migrations? Was that the restoration effort itself? I'd certainly like to think so. I'd feel, that would make us all feel good about the kind of work that we're doing in sort of coastal conservation efforts. But without long-term monitoring, without more data prior to that and after that, it's really hard to know. We've got a lot of plans for going forward, how we could do some of that long-term monitoring. I'll use Pleasant Bay again as an example, but here's an example of an important coastal conservation project that has sort of downstream implications for the open water system ahead of it, outside of it. But we need more data. We need to take a really comprehensive look at projects like this to know the effects that they're having on a broader scale. Staying in Pleasant Bay, I'm really excited to announce a new project that we're doing, very much in the context of figuring out what we need to know, what we already know, and how to monitor changes going forward. This is a project that's being done in collaboration with Sarah Griscom, the science director at Pleasant Bay Community Boating, and working in partnership with the Pleasant Bay Alliance as well. We're receiving funds from the EPA through the Restore America's Estuaries program, the Southern New England program in particular, to have a mini conference that we're going to have in the coming year. It may well be virtual, but it'll be a mini conference kind of looking at the state of science, the work that's being done by a variety of different organizations around Pleasant Bay. So those of you in the conservation community, you'll be hearing more about this. You'll be hearing from us looking for presentations as well. And from that, we want to build a working group that will help develop a framework and a strategy for long-term monitoring of the system based on all of the information that we've collected so far. Not only the work that we've done at the Center for Coastal Studies, but the great work done by the Pleasant Bay Alliance and by so many other organizations working around Pleasant Bay. And the idea is that we'll meet, again, probably virtually for at least the first part of the year, but come out of this with a plan for long-term monitoring and recommendations for future work. And of course, I wanted to close on this note. As Steve mentioned, we're kind of doing some of this in a sort of microcosm of sorts, working out on Simpsons Island. As Steve mentioned, I'm one of the board members of the newly formed Simpson Island Trust. I'm working with Erin Hilly, who's chair of our land management committee, and several other scientists and naturalists on a natural resource inventory for the island. And this is something that you want to do in the context of land conservation. It's a necessary part of any conservation work that we're doing. But for us, it poses a lot of unique challenges. This is an island with diverse habitats. You can see all of the different habitat types out on Simpson's Island there to the right-hand side. And of course, we're also going to be doing some restoration work on the island. We want to hope to monitor its recovery. We also have humans going out and doing things on the island, visiting the island, enjoying the island. So we want to be able to track the effects of that human activity in our restoration work. And so that's a real challenge. It's exciting to be doing this work. And from my perspective, and from the perspective of the trust, we are interested in research as well. It's part of the trust's mission. And so, on that note, a lot of these same principles apply at a finer scale here out on Simpsons Island. And I know there's a lot of folks here who are really excited about this project in particular, who've supported this project. And so stay tuned on that, because it is sort of in many ways, not only is it the heart of Pleasant Bay, as many folks like to say, but it's also a sort of a microcosm of a lot of the work that we're talking about here on a larger scale. We do see its potential for research as a long-term monitoring site, and essentially, as a living laboratory in partnership with a lot of the other scientific organizations working around the bay. And I think I'll close right here. I always like to end with a quote. And this is one of my favorite quotes. Terry Pratchett is he's a fantasy writer, novelist, and he says this very well in the context of baseline data, long-term monitoring, and where we go from here. If you do not know where you come from, then you don't know where you are, and if you don't know where you are, then you don't know where you're going, and if you don't know where you're going, you're probably going wrong. And that's a great way to summarize why it's important to monitor these systems, to get baseline data, and to go forward from there with the knowledge that we're all working together to gather. So I think I'll close on that note. I'll stop the screen share here. And Steve, are you going to moderate the questions we have here? Or how do we do this?
A couple of good questions coming in, and that was an excellent presentation, Owen. Thanks so much. So let's see. Judy Scanlon asked, did you find any winter flounder in Upper Pleasant Bay? Specific question, but I would expect nothing less from Judy Scanlon.
Great question. Without having all the data in front of me, I know we found some in Lonnie's Pond. And that's one of the ponds where they used to trawl for them back in the day. Again, all juveniles though. But yeah, that was probably the northernmost place in Pleasant Bay that we saw the winter flounder, was in Lonnie's.
And let's see, I guess this is a question for me and OCT. How will the OCT use this information? Will it influence future land protection prioritization? So that's a great question. I can say that we already prioritize land protection that is within the Pleasant Bay watershed, Nauset Estuary watershed, very highly. So that is kind of incorporated in our existing prioritization criteria. And I guess even more immediately, I'll plan to use some of these fun facts that Owen shared on our guided walks that are around the bay and elsewhere. But yeah, it's a very connected system. So we're always thinking about where our work is most useful, where we get the most bang for our buck, so to say. Let's see, what can we do ourselves to manage green crabs?
Well, that's a great question. I'm as a scientist, I don't tend to advocate for specific management practices, but I can tell you what they're doing up north in New Hampshire, in particular, is they're really trying to promote them as food. There's a lot of ways to cook up green crabs. There's no sort of fishery for them down here, but they are edible, and there's more and more work being done from a sort of a marketing perspective to encourage people to eat them, sort of eat the invasives, and that's one of those sort of candidate species for that. So that's one approach.
Let's see, do you study Sarah's Pond or Meadow Bog Pond? So Sarah's Pond, I know, is freshwater. Meadow Bog Pond, I think, is brackish. I know that both of those are looked at, I think, I know the Orleans Pond Coalition has been very involved with Sarah's Pond. And then I believe Meadow Bog Pond does have occasional water sampling done. There are people on this call that know better than I. So if any of you want to post in the chat, I can try to circle back to that one. But Center for Coastal Studies is not involved, correct, Owen?
No, we didn't go into either of those spots. There's so much more we could have done. It's such an incredible place to be. But yeah, there's a lot of places that we didn't get to.
Great. Kevin Galligan is asking that you comment further on the finding of mostly juvenile versus adult species of fish, and also long-term and short-term implications, causes, guesses of any of those findings.
Yeah, I mean it's a big question with not necessarily too many answers. It's some species we know more than others, know about more than others. For example, the winter flounder. There's been a lot of tagging work done in recent years, and what we're seeing is as waters warm, species like winter flounder generally are staying out of the estuaries as adults. They're spawning in deeper water, cooler water. They're not coming up into the lagoons and the coastal ponds like they used to. There's a lot of folks that think that's associated with the warming water temperatures. That fits a lot of the changes in distribution we've seen of other species. But in a lot of ways, I think the changes from adult to juveniles does say something like that for some species. But the fact that overall there's so many juveniles in there, that's a good thing. I mean, it's good to have nursery habitat, and it's habitat worth conserving and protecting. So I guess it's really species by species. We could say different things depending on what changes we've seen and how meaningful we think those changes are. We're still comparing snapshots in time. So although at least in the case of things like winter flounder, fishermen would tell you the same thing.
Okay, can you talk a little bit more about the drone monitoring technology and any of the newer technologies you're using? And also, separately, how has citizen science and working with fishermen played into long-term monitoring?
Yeah, I can't speak too much to the drone technology. It's literally and metaphorically out of my wheelhouse a little bit. I mean, we could. Uh, I don't know if anyone from Coastal Studies is on the call here who could talk more about that. But we have a whole team of folks, including several who are certified to fly them for research purposes, and but I can't speak too much to the technology. What I can talk a little bit about sort of the role of citizen science and the importance of working with the fishing community. And every one of these studies that I've talked about tonight, as well as some of the other work that we're doing, has really been, it's been informed by the fishing community. Fishermen participate in a variety of different ways. If they see something new, they come and tell us about it. If they, in many cases, we think we found something new, and they'll be like, no, we've been seeing that for a long time. So in a lot of ways, it helps us contextualize some of our observations and think about which things might be more important to look at. But fishermen, because they're on the water so much more than we are, they're often the ones who see change first. And so in the context of sort of fishermen and citizen science and long-term monitoring, those folks who are out there all the time are the ones that see the changes first. So it's important to listen to them.
Kind of along those same lines, are you noticing more blue crabs? And are these the Chesapeake crabs? I think the answer is yes to that. And then are you making any guesses about whether this is due to global warming, as they might be shifting northward in their range?
Yeah, that's a great question. I mean, we're certainly seeing blue crabs all over the place now. And there is some evidence to suggest that could be due to warming waters. That's fairly typical for shifts in species distribution. Um, I know it was very unusual to find very many blue crabs around the Cape a few years back. There was always those particular spots on the south side there, but they're popping up more and more in Pleasant Bay, Nauset, around in Cape Cod Bay. We caught one in a sea scallop dredge in Cape Cod Bay not that long ago. So they are in more places, and we're seeing that they're sticking around. We've seen egg-bearing crabs. We've seen the little tiny juveniles like the one you saw on my finger there. So it certainly looks like there are more of them here. Whether that's due to warming waters, it's hard to say, but I think that's a reasonable guess. Again, each one of these species, there's so much more information out there. There's probably someone who's already doing that work and could answer that question better than I am. Very, very much a generalist here with a lot of this stuff.
Okay, this is a good one. What's the best way to get involved in this work? Is the Center for Coastal Studies still in need of any volunteers for this kind of thing?
Yeah, we still do have a volunteer program even during these times. Obviously, it's much, we're working in a socially distanced way in a much diminished capacity in terms of the number of people we have involved in a lot of our activities. But please do submit a volunteer application if you're interested in helping us out. We have taken on some new volunteers even during these times. There's opportunities to work outside in the wonderful weather that we're having now, and do field work. So yeah, please do let us know. You can go right to our website, and there's a link to email for volunteer applications.
Great. Are you aware of any studies that have tracked the boundary changes of Pleasant Bay, Nauset Marsh, or Town Cove, and where might someone be able to look into those findings of the shifting boundaries?
Yeah, I guess I'm not 100% sure what you mean by shifting boundaries. I know we've seen, and I'm not sure this is what what you're getting at here, but we see sort of latitudinal shifts and sort of there used to be a little bit more of a distinction between species communities between what you see in Pleasant Bay and Nauset, even though they're relatively close to one another. And there's a PhD student at URI a number of years ago from Wellfleet, um, Ethan Esty, who did his doctoral work looking at kind of where that change point was between species communities along the eastern shore of the Cape. And it'd be interesting to kind of revisit that as we get more data from Nauset and see how similar the communities are between Pleasant Bay and Nauset. So maybe we could tell you more about that after we do another several months of survey work.
I think I interpreted that to be a question about the actual, I guess, shifting sands and channels and that kind of thing. So is there a good resource for seeing those shifts over time?
Yeah, I mean, I know that in our report, at least on Pleasant Bay, we show a lot of what we do know about the changes over time from historical information. I'd gladly take an email after this and connect you with our geologists, because I know they have much more comprehensive information and historical maps and could probably very easily point you to that. And I think some of that might actually be in either the Pleasant Bay Natural Resource Management Plan or the coastal atlas that's out. If you send me an email, I can dig around real quick and see if I can find that. I don't know how far back some of that stuff goes. I know we have some records that go back to the 1800s from our geology department. But.
Well, and we actually tried to get Graham Geist to do one of these lectures for OCT, but unfortunately that was early days of Zoom and he wasn't comfortable with the platform. So here we are, stuck with Owen, unfortunately.
We've just started seeing Graham on camera during our staff meetings, so we're getting there. We're getting there. You might try again.
Okay, where are the older fin fish? Do they escape to the ocean, die, or are they fished out?
Great question. Again, I think that's sort of very species specific. The winter flounder example is one that we know about. But again, like a lot of these species are pelagic species that tuck into a place like Pleasant Bay for a short time in their early life history. It's a good sheltered, protected habitat for them. So things like your sea herring or the scup or the sea bass. We even saw some mackerel in there recently. And those fish are pelagic. They're more offshore. That's where they spend most of their lives. But when they're small and more vulnerable to predation, they like habitat with more structure in some cases. There's a lot of food for them to eat in these estuarine and coastal habitats. So there's a lot of reasons why the small ones are there. But these in many cases are species that spend the rest of their lives offshore in deeper water. Every species has a slightly different life cycle, and some of them are changing over time.
Okay, just to circle back to an earlier question, we did get confirmation that Orleans does sample in Meadow Bog Pond. And then there are a couple of questions about eelgrass and the extent of eelgrass in Pleasant Bay, whether anyone is tracking any change of that and whether you've seen any change over time as to the distribution of eelgrass.
Yeah, I don't have that handy. I know it is changing, and I know we looked at it a fair amount with our seafloor mapping work. The state looks at it. There's a lot of different groups studying it out there. We've certainly seen changes in the eelgrass over time. As far as a sort of a number, an acreage and how that's changed over time, I don't have that handy. I know we observed a lot of changes even during the years that we were studying it. Some instances where sand waves were actually smothering some of the eelgrass beds. We saw that on sonar. So we saw changes even during our studies. And we certainly mapped a lot of that out with our sonar work. But there are other folks that could probably answer that question a little better than I could.
This is an interesting one, and I think you're going to need to explain to me, Owen, but is there an existing system for managing black mayonnaise and restoring that bottom through dredging or other means? Any plans for Pleasant Bay? And that's from James Rosado.
Oh, great question, James. it's funny. We just wrapped up a study in Wellfleet. Agnes Mittermeier, who's our benthic ecologist in residence at the center, she did some work with it, and we've actually renamed it Black Custard now. Apparently, that's a whole other story. But the short answer is no. We at the center, that's not something we've really looked at. I've heard some ideas about how one might repurpose that black mayonnaise for other uses, maybe extract it from the system and do different things with it. I've heard some different ideas about that, but I haven't heard any sort of management level conversation from a municipality or natural resource agency. Just some really interesting ideas from the community.
Well, the questions just keep coming in, which speaks to your knowledge base, Owen, and all the curiosity you have generated in people. But I'll ask a couple more here. We'll go for a few more minutes, and then maybe people can circle back with Owen via email or elsewhere to get some answers. But let's see. Have you taken into account the changing break in the barrier beach in comparing your results to those of the 1960s?
Yeah, I mean, essentially, that's one of the reasons why, as I tried to allude to a little bit, is that it is hard to compare those two things, because there are a lot of differences. There are fundamental differences in the barrier system, in water quality, and so many different things. So with that question is a great justification for having that long-term monitoring, because without that, it's hard to understand how those changes affect the communities over time. So if you have two snapshots, it's like a scatter plot with 2 points on it. It doesn't really tell you very much until you can see how they all lay out. And so, yeah, the short answer is no, we didn't and couldn't really take that into account, and we need to.
So we've got a couple lightning rods here. We've got one about dredging and one about seals. So do you want to take dredging first, how that might impact fisheries habitat? Although I know that's a massive question that is very much being looked at.
Yeah, I think I'll leave it to the folks who are looking at that to answer that one. In all fairness, it's not really my area of expertise. I mean, we can certainly document changes, but that's a little outside my wheelhouse as far as I collect a lot of that data. I share it with the folks who are asking those questions, but yeah, it's not something I can easily answer. It's a complicated question.
And then I know that you didn't specifically talk about this, and I don't know whether this is outside of your area of expertise as well, but can you comment on seals and how they might be impacting fish and invertebrate populations in these estuaries?
Yeah, I mean, we did, when we worked in Pleasant Bay, for example, we looked at seal populations, we looked at seal diet in those areas. As you'd expect, there was some overlap between what we saw in the fish community and what the seals were eating. But as we've grown to expect, a lot of sand eels and other species in their diets. We saw the most small winter flounder around where the seals were hauled out, which is interesting. But seals choose their haul out sites not necessarily based on food availability, but due to access to the water. So I did think it was interesting that despite the number of seals, there were so many small winter flounder there. But again, these are sort of almost anecdotal observations. It's really hard to answer that question in a really rigorous way, except to say that they're there. They eat some of the things that we saw. They eat a lot of other stuff too, and that's about the long and the short of it. There are folks looking at things like water quality and how those might be affected by seal presence. But again, without a longer time series, it's hard to interpret what we see.
Mm-hmm. What are the highest impact actions that a community can take to implement or can take to support the fisheries habitat?
Ooh, great questions. Again, being a scientist and not a resource manager, sometimes it's hard to answer those kinds of questions. But I think one of the things that comes to mind is really continuing a lot of the great work that's been done to improve water quality, to reduce nitrogen inputs. The Pleasant Bay Alliance in particular is doing some really great work with water quality around Pleasant Bay. I think that's a really important issue for habitats like this. Doing what we can to protect salt marsh habitat, eelgrass beds, a lot of these habitats where there's structure really important. Those are kind of the first things that come to mind.
That's great. Let's see, is there any analysis going on regarding bird life in these coastal waterways that CCS is involved in, or anyone else?
Yeah, we don't do a lot of work with birds. It is a key piece of the puzzle in a lot of places. And, unfortunately, we have a lot of expertise around here from Mass Audubon and other groups that do a lot of work with seabirds and shorebirds. But, it's not something we do a lot of, but it is a really important part of the system.
Okay. Curious about your thoughts on scale. Wonderful data sets on macro inverts and inverts, but how valuable are microorganism inventories? Does anyone in your organization have a skill set to apply a sequencing-based approach to ID these tiny critters?
Oh, great set of questions. I would say I'll answer sort of the bigger picture question first, and that is, we do see linkages between sort of the larger invertebrates and the communities of the smaller invertebrates that are in those sediment samples. And many of them also can serve as indicator species for habitat quality. There are some species that are generally associated with better water quality, or relative to sort of anoxic sediments and things like that. So there's a lot of reasons why those benthic organisms are important. The diversity of those organisms is often directly linked to species distribution and diversity of larger organisms. So there's a lot of reasons why we look at that. They can tell you a lot about the system. As far as a sequencing-based approach, that's not something we're doing right now. There's more and more work being done with environmental DNA, and it's a really exciting study area, but it's not something we're doing. We're still doing it old school in the sense that we're sieving samples, picking out microorganisms, and staining them pink and counting and identifying them. We do it the old-fashioned way.
Good, clean fun. All right, I think I'm going to do one more here, and then we will have gotten through at least the large majority. So thanks again, Owen, for your patience here and thoughtful answers. Let's see, we seem to have much more plant growth in Meeting House Pond than a few years ago. Any insights on that?
Yeah, unfortunately, it's not an easy question for me to answer. We haven't been back there in a few years ourselves. I would probably defer to the folks that are doing water quality monitoring in that area. They would probably be able to answer that question better than I.
And I should note that Orleans is always looking for volunteers to help with that water quality monitoring, as that has come up a few times tonight. So, well, great. Thank you, everyone, for being available tonight, and special thanks to Owen and everyone at the Center for Coastal Studies for all the great work that you guys have been involved in for many years now. And we're just so happy to have an organization like CCS in our community, so OCT can focus on the land and let CCS do their great work and educate our members at opportunities like this. We had over 80 people joining us at one point, so that's great. We're a big draw, Owen.
Well, it was an absolute pleasure being here, and I hope we can work more together at that land and water interface over time. And please do, if there's a way for you to share my contact information with the attendees tonight over email or something like that, you're more than welcome to share my email. I'm happy to answer any follow-up questions. So I wish I could see you all in person, but it's a great group with great questions. So thank you so much for having me tonight.
That's awesome. Well, thank you, Owen. And I should note that Owen gave us permission to share the recording of this webinar on our website. So if there's anybody that you thought of during this that you want to share the presentation with, that will be up on OCT's, the Orleans Conservation Trust's website in short order.
Great.
Well, thank you again, Owen, and thanks everybody for coming out. Have a great night.
Pleasure.